Biotech News Television. I am Jennifer Herndon.
And I am Kayla Lashley. Today you're going to learn about everything from glow-in-the-dark cats to singing mice.
We'll begin today by talking about glow-in-the-dark cats. In 2007, a South Korean scientist altered the DNA of a cat to make it glow in the dark.
He then took that DNA and cloned other cats from it. Can you believe this right now?
No kidding, Kayla. This is exciting news.
Our sources tell us that researchers took skin cells from Turkish Angora female cats and used a virus to insert genetic instructions for making red fluorescent protein.
Next, they put the gene-altered nuclei into the eggs for cloning, and the cloned embryos were implanted back into the donor cats.
This made the cat the surrogate mothers for their own clones.
Talk about reproduction. Now we move on to why scientists would go to such lengths to produce these cats.
Scientists say that the ability to engineer animals with fluorescent proteins will enable them to artificially create animals with human genetic diseases.
Therefore, the pros of glow-in-the-dark cats include that they allow scientists to have the capability to see genes that work to make the cat immune to certain diseases, based on whether the cat glows or not. It could help with the treatment in cats just as much as it could help with the treatment in humans.
Let's move on to the cons of glow-in-the-dark cats. This includes that they are unknown side effects and it could potentially be a waste of money.
And that wraps up our discussion of these up-and-coming cats. Now we'll go to Brooke, Paige, and Sydney for their discussion on spider goats.
In today's news, we're here to learn about a genetically modified goat.
Now, throughout history, the goat has been one of the most adorable livestock on the farm. Recently, biotechnologists have been studying how to incorporate the DNA of a spider and a goat.
Could you picture a goat squirting out a spider web? Well, you don't have to, because that's not exactly what happens. The goat will still produce the same milk they always do, but it will be enriched with spider silk. So it's new and improved. Applications of artificial spider silk could include using it for artificial ligaments and tendons due to its elasticity. And it is also a natural product, so it will work well with the body.
For this to occur, scientists take DNA from a spider that creates the silk web and inserts it into a goat's DNA. The silk from a spider is very durable. You may wonder why anyone would want a goat that produces spider silk. The milk that contains the spider silk will be a lot more stronger than regular milk.
This creates many benefits to the cheese, yeast, and other products that can be produced with a goat's milk. It's quite interesting the way that scientists can take DNA from one animal and insert it into another.
The silk that the goat now has is used for synthetic fiber. This material is used in everyday items such as tires, rubber products, and protective gear.
Who knows what is going to be next in terms of genetic modifying? Currently, there are 30 spider goats at a university-run farm. As long as they never make flying spiders, I think we will be okay. Thanks for joining us. Now, to learn about the BioPig, here's Sydney Yarborough, Zoe Coleman, and Breezy Castle.
Thank you, Sydney, Paige, and Parker. I'm Zoe Coleman.
I'm Breezy Castle.
And I'm Cindy Yarborough, and today we're going to tell you about the EnviroPig, or as many like to call it, Frankenstein.
For those of you who don't know, the EnviroPig is the newest phenomenon today.
Today, we'll tell you the who, the what, the when, the where, and the why.
So what is the EnviroPig? Let me tell you. The EnviroPig is a Yorkshire pig that is genetically modified to better digest and process phosphorus. This EnviroPig was released publicly on February 20th, 2010 in Ontario, Canada, when the Canadian government determined the EnviroPig could be produced outside of research context and controlled facilities.
Most of you are probably wondering why we need such a thing as the EnviroPig. Here's your answer. Typically, Yorkshire pig's manure is high in phytase, a form of phosphorus. So when farmers use the manure as fertilizer, the chemical enters the watershed and causes algae blooms that deplete oxygen in the water and kill marine life. This phosphorus is derived from the corn, soybeans, and barley that the pigs eat.
The creators of EnviroPig say that by creating the EnviroPig, they are able to digest phosphorus and phytic acid found in their food by producing an enzyme in their salivary glands. Hence, there's no need to include a mineral phosphate of commercially produced phytase to balance the diet, which is normally carried out in pigs. EnviroPigs help improve the environment as their manure does not contain high levels of phosphorus.
How is that done, Sydney?
Well, Breezy, scientists combined a bacterial gene from the enzyme phytase with a mouse's gene that controls the production of proteins. Then, those genes were inserted into a fertilized pig embryo and inserted back into the female pig. After 114 days, the baby pigs were tested for transgene and phytase enzyme activity in their saliva.
So what are the benefits to EnviroPig? EnviroPigs reduce pollution by up to 60% and eliminate phosphorus pollution in lakes, streams, and ponds. They also reduce the energy requirement for meat production.
But it also has cons. Some people have not approved the eating of EnviroPig's meat because they may have allergies in the meat that could cause illnesses. EnviroPigs could potentially be costly.
Wow, some pigs, huh?
They sure are. Next we'll hear from Michaela and Carly about singing mice. Stay tuned.
Thank you, Zoe. Today we will be talking about a new discovery in biotechnology known as singing mice. Scientists in Japan have genetically engineered a mouse that tweets, no, not like on Twitter, but like a bird. Contrary to what you might think, these mice were not made by putting bird DNA in them. Here to tell us more is our field reporter Carly, who has been researching the experiment.
Thank you for having me in the studio, Michaela. This is a discovery to confuse kindergartners worldwide that are trying to learn their animal sounds. But you're correct. These mice are all part of a bigger evolved mouse project at the University of Osaka, in which mice DNA is being altered so that it's more susceptible to mutations. They have been crossbreeding these mice for generations to observe the outcomes.
Now, what I've been told is that the discovery of these mice was accidental. That when checking one litter of baby mice, the scientists noticed that one was not making normal mouse sounds, but tweeting instead. Is this information correct?
Sure is, Michaela. But since the firstborn, the labs have been able to reproduce over 100 singing mice to study. The hope is for them to learn more about human dialect evolution, so they mix groups of ordinary and singing mice together to see how they still communicate beyond the language barrier.
Since they are studying language and sounds, is there a reason why they couldn't use another animal, such as songbirds?
Well, many other countries tried that in the past with songbirds, but this particular study uses mice because they are mammals with genetics much closer to humans. Even with that said, a con of these experiments is that they cannot provide exact results for why humans' language evolves, because these are mice, not humans.
But it is still one step closer to new findings and knowledge. Now we will play a video of these mice so you viewers can see the developments for yourself. Thank you for watching, and now we're going to turn it over to Hannah and Caitlin for a report on banana vaccines.
Thanks, Michaela. How would you like a tasty banana that has a vaccine in it? Well, guess what? It may be coming soon. Bananas were developed through genetic modification that offers vaccines against diseases such as cholera and hepatitis. Just like with the needle vaccine, people who eat them develop disease-combating antibodies that make them immune to a disease.
Most likely, these vaccines would be grown in a controlled environment where cross-pollination and adverse effects on other species would not be possible. But, considering what would be the widespread use and disposal of these foods, there is potential that the gene could enter domestic food supply. So ingestion in large quantities and ingestion by certain people with health conditions may be very harmful. There also is the primary issue of effectiveness of modified foods as a vaccine.
Charles Arntzen, a plant biotechnologist at Arizona State University, had in mind producing bananas engineered to make a protein from the bacterium that causes cholera.
This protein doesn't cause diseases, but it's a very good vaccine, Arntzen said. The bananas would be harvested and made into chips and powder, and these would be given to people in developing countries who don't have good access to modern vaccines. The banana chips, once eaten, would expose the body to cholera protein and offer resistance later on to encounters with the cholera bacterium.
The cost of producing edible vaccines will be as much as 10 to 50 times lower as compared to normal vaccine production. Another more obvious advantage is that syringes will not be required, which allows vaccinations to be less painful and more importantly reduces the risk of transmission of infectious diseases. which run rampant through third world countries. Instead, the vaccines can be delivered by food products like bananas, which can be eaten raw and served in a puree form.
And now to Caleb and Peyton, who will talk about genetically modified trees.
Thank you, Hannah and Caitlin. We're here to talk about genetically modified trees. A genetically modified tree is a tree whose DNA has been modified using genetic engineering techniques. In most cases, the aim is to introduce a novel trait to the plant which does not occur naturally within the species. Examples include resistance to certain pests, diseases, environmental conditions, and herbicide tolerance. Genetically modified forest trees are not yet approved for commercial use, with the exception of insect-resistant poplar trees in China.
The trees grow faster with the genes that the scientists have put into it. Several genetically modified forest tree species are undergoing field trials for deregulation. Much of the research is being carried out by the pulp and paper industry, primarily with the intention of increasing the productivity of existing tree stock. Certain genetically modified orchard tree species have been deregulated for commercial use in the United States, including the papaya and plum.
The development, testing, and use of genetically modified trees remains at an early stage in comparison to genetically modified crops. Research into genetically modified trees has been ongoing since 1988. A precondition for further commercialization of genetically modified forest trees is likely to be their complete sterility. A precondition for further commercialization of genetically modified forest trees. Plantation trees remain phenotypically similar to their wild cousins in that most are the product of no more than 3 generations of artificial selection. Therefore, the risk of transgene escape by pollination with compatible wild species is high.
Alright, one of the most credible science-based concerns with GM trees is their potential for wide dispersal of seed and pollen. Because of these intangible values and long lifespan of trees, it is impossible to treat forest as a commodity within an agricultural model. The gap between forest and agricultural systems has become especially clear in the context of genetic engineering. The successful introduction of genetic engineering in agriculture for a limited number of traits and species, promoting forest scientists and managers consider its use as a management of production tool in forestry.
This subject has generated heated debates and violent reactions which have often lacked the support of objective information. Furthermore, the existing scientific information has been contradictory, allowing for questions concerning its credibility. The competitive zeal of some biotechnology owners has added more fuel to the debate.
Alright, now we turn over to Jesse Morris and Ethan Warren with their growing genetically modified salmon.
Hey, I'm Ethan Warren and this is Jesse Morris. We'll be talking to y'all about fast-growing salmon.
On today's news, there has been a new discovery by scientists of a U.S. company called Aqua Bounty Farms. They have found that through genetics, they can create a salmon that grows 2 to 4 times faster than any other salmon. Everybody knows that salmon do not produce growth hormones year-round, but scientists have made a modification that allows them to grow twice as fast. Scientists can keep this hormone active by using a gene from an eel-like fish called an ocean pout. There is no difference in the fast-growing salmon and the regular salmon besides the size. The texture, flavor, odor, and color are the exact same. Although they have similar characteristics, there is a problem with these fast-growing salmon. They might not be safe to consume.
Good point. This is a debate that has been long talked about. If these fish are approved by the FDA, then it will be the first time the government has allowed for modified animals to be marketed for human consumption. This leads to another great debate because even if this salmon is approved, it would not be labeled as genetically modified. Many people believe that modified organism used for human consumption should be labeled so that way they would know what they are eating.
There have been studies that genetically modified salmon contain a 40% higher level hormone that is known to cause cancer. The genetically modified salmon exhibits as much as 52% higher levels of allergen potency, whatever it is, which means that there could possibly be a chance of an allergic reaction from consumers. The GM salmon contains 5% less protein and 58% more fat than regular salmon. There are many pros to producing this salmon, such as the size, but based on the numbers, should we really eat this fish? That's today's news. Now to Jonah Mayo and Josh Moore reporting on what seems to be super cow.
Good morning, I'm Sarah Michael.
And I'm Kayla Haley here with you this morning.
We'll be talking about less flatulent cows and how they affect our world. Less flatulent cows are cows who are genetically engineered to produce less amounts of methane in their digestive process. Gases from flatulent cows contribute to global warming. Agriculture research scientists from University of Alberta found that bacterium is responsible for producing methane and designed a line of the cow that creates 25% less methane of an average cow. Methane is a major contributor to the greenhouse effect, so scientists are trying to genetically engineer a cow to produce less methane. A cow per year releases from between 70 to 120 kilograms of methane. How does this happen? Well, scientists put the cows on a diet off of omega-3 and omega-6 acids called the Vorlex Blue Blanc Core Nutrient Program. Scientific research shows that the balanced diet could produce more methane per cow. To protect our climate, French farmers were giving the cattle herds feed that would reduce how often the animals produce methane.
In research, it says that in 2010, the UN made a global levy on the methane emissions. Something similar to this was proposed to the UK 3 years earlier. Methane is produced by cow flatulence and has an effect on global warming. Cows that fart less than average have been produced to fight the effects that cow flatulence can have towards the environment. They do this so that new breeds of cattle on the farm would produce less methane. Australian environmentalist Tim Flannery says sustainability is vital for the long-term survival of the farming industry. Limiting the flatulence of methane gases not only has a positive impact towards the environment, but it could increase the productivity of the animals.
That's all for today, folks. Thanks for watching the Biotechnology News. We'll see you next week for genetically modified dinosaurs.